The goal of this discussion is to give you some practical, maybe a little bit of simplified overview of some of the concepts that are talked about in the Svensson and L. Mabrook paper. That's paper 15. Like many papers in this course, it can be a little bit technical. Again, if you find yourself getting weighed down or it's too complex, the body of it, skip over those parts, skim over it. Read the abstract, of course, maybe part of the introduction and then the discussion, and then come to these lectures, these discussions, and we will give you the details, the information that you need to pull. So if you're getting overwhelmed by those papers, don't worry too much about it, and don't spend too much time on them. So I'm going to review some of the ideas and concepts. Some of these are explicit in that paper, and others were kind of taken for granted. So in this diagram, I have three species, and we're going to assume that we have accurately reconstructed the phylogeny of these three species. And so the big black tree, the kind of larger tree, represents species relationships for species A, B, and C. Now within this, I have drawn thinner lines that I've color-coded. And these represent a gene history for a single gene that went through a duplication event, and I've tried to mark those very clearly. We have a gene duplication event. They're represented and color-coded, the two different paralogs, right? So that's a good vocabulary word. Let's actually add that down here. I'm assuming you already have that, but if not, that's okay. Right, so we have two types of orthology that we are going to be talking about here. We have paralogs, right, which are two genes that can trace their history back to a duplication event. That's where they get a common gene ancestor. And then we have orthologs, which are two genes that can trace their common history back to a speciation event. So when we have these genes breaking into two descendant genes at the same place where the species break up, that means they're orthologs. And the color coding helps too here, right? So two genes that are green are orthologs. If we have a green and a red, they're paralogs. So in this case, the color coding is supposed to help you, okay? So make sure you're familiar and comfortable with that definition that they are two subcategories of gene homology. All right, so this is where it can get a little bit complicated, and I've purposely done this to show you how this idea of gene duplication and paralogy and orthology can make our gene history potentially different than our species history if we don't understand the complete history of the genes. Now in these diagrams, it's as if we're able to see everything occurring in the past, but in reality, we have to use some sort of a method, an approach to figure out the gene and species history, and so there may always be some amount of uncertainty, particularly if the data we're using to reconstruct gene history and species history are limited, or maybe are subject to some of these factors that can make reconstructing those histories a little bit difficult. But for this example, we're going to assume that we know explicitly the history, okay? So we're going to start here with a single ancestral gene represented by this green line, and then we have a gene duplication event, okay? And so we end up with two copies of that gene. Now, gene duplication events can occur via a number of mechanisms. One of the most common mechanisms, oops, let me get this bigger. That's the one I wanted. One of the most common mechanisms for a gene duplication event is when we have a mistake made during meiosis, when we're actually a little bit prior to meiosis, when we're going through the copying of the DNA. And a mistake is made, so we copy a section of the gene twice. This can also occur through unequal crossing over and often results in many genes that we get duplicated. And this is like any other mutation. It can be good, it can be bad, it can be neutral, and is subject to the forces of natural selection and genetic drift. So we are assuming here that whatever happened, this was a beneficial or at least a neutral mutation and eventually became common so fairly soon after it occurs, maybe thousands of generations, but geologically that's a rather quick period of time. And so soon every member of the species carries two copies of this gene that can be traced back to that ancient gene duplication event. Now, one other little concept that I want to talk about, and then a related term that is brought up in the Svensson and Mabrook paper. Okay, this is anagenesis versus cladogenesis. These are two ideas that and definitions that you should know from evolution. Hopefully you remember them. If not, let's review them. Anagenesis refers to a change in function over time, but without any new genes being created. Okay, so we can have anagenesis occurring where genes change in function. Cladogenesis refers to a speciation process, making new clades or new groups. So typically when we think of evolution, we think of it as a combination of these two things, of species breaking into new species and then differentiating over time, and that differentiation occurs in their genes and is anagenesis. Okay, so here the gene duplication event is really a type of an anagenesis where we have more variety and diversity. And often what can occur when we get a duplication event, and this is the power of gene duplication in evolution, is we can maintain whatever the ancestral function of this gene was. Okay, so let's say that this is a hemoglobin gene, and so it's carrying oxygen. And then maybe we get a second copy that allows them to work together in a new way that can carry oxygen more efficiently. So we maintain the ancestral function of carrying oxygen, but maybe add on to it. And so I'm going to add a couple more vocabulary words here. And we've got these, at least some of them explicitly and some more implicitly. But we've got these diagrammed here, okay? A pseudogene refers to a region of DNA that loses function as a gene. And it's still there. So notice I've got gene loss. What I mean here is most likely we didn't just lose that section of the genome, although that can happen, but that's rare. More commonly what occurs is we get mutations occurring in that gene so that it is no longer transcribed and translated. So it's part of the genome still. We maybe could even search it if we did an entire genome sequencing project. But we cannot identify it because it's not transcribed, so it doesn't have a protein product. So it really no longer is a functional gene, but we may be able to identify it from its sequence. So if we're only looking at the genes that are being used, we're going to miss all of these genes that have had mutations occur either in their DNA sequence or, of the protein itself, the protein coding region itself, or maybe even in the control mechanism, so it's there but no longer turned on, it's no longer expressed. And so this would be referred to as a pseudogene. Oops. And we have many examples of pseudogenes, and now with entire genome sequencing projects, we're much more able to identify them. But in the past, that wasn't so. We were much more keyed in on looking at these genes that were being expressed and used. Okay. The last term that I want to do here relates directly to anagenesis, and this is called Neo-Functionalization. Big vocabulary word, but nicely, if you understand the components, it makes sense. Of course, neo meaning new, novel, function, of course, referring to the function of a gene. So neo-functionalization is where anagenesis makes a gene take on a brand new function that it did not have in its ancestral form. And so we can have both of these processes occurring. Now, I have purposely drawn this example to illustrate a point. Notice that if we had 3 separate independent gene loss events, that if we came along and were unable to identify the pseudogenes in the genomes of these organisms, if we only looked at the genes that were being used, we would get a misleading assessment of the history of these species if we were only using these genes. So notice that 1 and 2 are orthologs of one another. They're green. And so they share a common ancestor right there. Let me get my little arrow here. Right there is the common ancestor of gene 1 and 2. And that's why they're orthologous, because their common ancestor split, the gene ancestor split when the species split. But notice if we throw in gene 3 here, we trace it all the way back here. Its common ancestor is separate, the red line, separate from these ones, common ancestor. And so we'd have to trace it even farther back in. Boom, there is the common ancestor for all 3 of these genes. And if we looked at the gene history of these 3 organisms, using genes 1, 2, and 3, we have a combination of orthologous genes and paralogous genes. And so our gene history for genes 1, 2, and 3, if we reconstruct it accurately, looks something like this, right? Where we have genes one and 2 that share a more recent common ancestor. I'll put the numbers in here. So genes 1 and 2 share a more recent common ancestor than genes 3. So notice in this case our gene history, because we have a mix of paralogs and orthologs, doesn't match our species history. So that's just one example. Now, if we understood these pseudogenes and we're able to identify them and sequence them, we may be able to include them and accurately reconstruct. So if we include all of the genes that have occurred, what we will actually see is something that looks like this, where we have the species phylogeny represented twice. And if we reconstruct everything, we have all of the information, we could potentially reconstruct the entire history that would look something like this. Okay, so we would have the species history. I've ran out of space a little bit here, but it's not too bad. Let's just make that up a little bit higher. Okay, so let's put numbers in on these genes so that we can see this more clearly. I'm going to add, and the numbers are just random. They're not supposed to mean anything other than a way to identify the different genes. So there's no rhyme or reason to why that one's 4 and the other one's 1. It's just so we can identify them a little bit better. Okay, so we have number 5. So we have 6 genes that we have identified when we have identified all of the working genes and all of the pseudogenes. And if we're able to then reconstruct it once we've identified it, what we would see is gene 1, 2, and 6 share a more recent common history, right? So it would look like this, and we would have genes 1, make them the same size, 2, and 6, right? And so this part of the phylogeny then reflects the green branch of the tree, right? So 1, 2, and 6, notice it's the same exact pattern and that it matches the history of the species. And then we would have kind of a duplicate image of this that would be the red part, 4, 5, and 3. And so we would have something like this, where we would have 4, 5, and 3. And of course, then this second side of the tree, the right half of the tree here, represents the red history, which we have kind of overlapped one another here. 4, 5, and 3. Its pattern is the same as the red branch over here, and it matches the species history. And so when we have a tree, a gene tree, it looks like a phylogeny, and really is, except it can contain both speciation events and duplication events because it's a gene tree rather than a species tree. And so notice that I'm just going to put let's put little circles around each of the speciation events. So there we have speciation event number 1, or maybe we should call it number 2 because it happened after the other speciation event, right? That is the same as that speciation event, right? That's when gene 2 and 6 broke away from each other and gene 5 and 3 broke away from each other when those species broke apart. And so those are speciation events. And then we have a second group of speciation events. Let's do a slightly different color. Okay, so here we have the first speciation event represented there and there in the gene history, corresponding to this speciation event right there. And then finally, in the bottom, in the basal node of this tree where the genes first broke apart, let's make this one a square because it's not a speciation event. So we'll make it a little bit different looking. So there's a gene duplication event that corresponds to this ancient gene duplication event right there. Okay? So you should be familiar and comfortable looking at these large phylogenies like this and being able to tell about the gene history and the species history. you should also know how gene loss, right, can lead to a confusing outcome if we don't consider all of the genes, maybe because they're pseudogenes. Now, today our data sets are bigger and we can do more sampling, and maybe we could throw in extra species and get a more complete picture to look at this. But anagenesis, if these genes change function, can make it difficult because genes 1, 2, and 3 might even have separate different functions or slightly divergent functions, so it's not completely clear based only on their function how they are related to one another. Now, I'm going to give you a real-life example to finish up. So, this is an example from amniote opsins. So just to be clear here, amniotes are organisms that have an amniotic membrane around the developing embryo. So this includes all of the reptiles, and birds are part of the reptile group, right? They have eggs that can lay on land and not dry out. And of course, mammals have adapted now. Most mammals, the monotremes like the platypus and the echidna still lay eggs. But most mammals have adapted to have an internal amniotic membrane, but it's still a really important part of embryonic development and aids in transfer of some vital components, but also protects the embryo and separates it from the mother while it's developing. So anyway, these are all amniotes. And opsins are genes that are expressed in the eye, and they are light sensitive. So they're critical. They're the primary way to transduce a light signal, electromagnetic wave, into a nerve signal so that we can have vision. Okay? So birds and reptiles in general have 4 opsins. And there's some exceptions to these rules. I've color-coded them, but just very generally, we're really giving you a very broad overview of this. Okay? So birds have 4 opsins. Almost all mammals have 2 opsins. But the hominids, right, which includes humans, chimpanzees, gorillas, orangutans, all have 3 opsins. So we have basically a short wavelength, a medium wavelength, and a long wavelength. And that allows us to see and distinguish a higher variety of colors. Now, there's some interesting things that do not really relate to this that you might have known about before. For instance, one of these opsin genes is on the X chromosome, so there's some sex-linked things that go on, and men tend to be colorblind more often than females if they get a bad copy. But we're not looking at that. We're looking at more big picture across this major group. Now, how did these opsins evolve? There are a number of ways that might explain it. So for instance, it might very easily be, and I'm going to draw in some lines here, it might very easily be that there were anciently a single opsin that went along, and then there was a duplication event. We'll color code these later. Oops, I don't want any fill. So let's do this. There we go. Okay, so we have a gene duplication event here. And so what that means is the ancestor of all of these organisms, there's our 2 genes now. In fact, let's make this one go up here. It makes it a little bit easier to get everything down the line. So somewhere down the line, that duplication event means that each of these organisms carries 2 genes into their ancestor, right? So we can see that. And with them all the black color, it's difficult to see. So let's highlight them so we can make sure that we know what we're talking about. Oops. I want to do that one. Oh, I see why. Okay, that should be a little better now. So let's make 1 copy of these genes red. Sorry, I want that one and then red. There we go. So we got the red ones, right? And then we'll do the green ones. And we'll just make the ancestral one green. And again, you might look at that and say, oh, maybe this copy has maintained the ancestral function. But color is not really supposed to designate function here. It's just to show you the 2 separate genes that have descended, 2 lineages of genes that have descended from that ancient gene duplication event. Okay, so ancestrally, we could assume that the ancestor of all of the reptiles, including the birds, had 2. The ancestors of the mammals had 2. And then in the mammal branch, we can just extend that. It's a rather simple story. So we can continue this here and say, well, maybe instead of red and green for these, let's change this one because it'll make it a little bit easier here. Let's change this one something more similar to that bluish purple color. Okay. All right. So we have this branch that connects into that gene. I might have to rearrange some of the orders of the way I put my genes here. Sorry. Shift, undo. Okay. So there we go. So there's that one. And then this ancestor still had 2 genes. Make it a little bit lower here. It's not going to be all pretty and symmetrical, but you'll get the idea here. And then, of course, this same gene continues up into the hominids, and so they have a red version also. And we'll just shift this over so that the colors match up. Okay, so now we have explained how that red one evolved and descended in the mammals. Okay, so we can do the same thing for this purple version. Okay, so now we have 2 copies of the gene ancestrally. At speciation, they break up, and all of the other mammals, I just put canines as an example, but this could include all of the other mammals. This is one reason why, well, this is the reason why your dog is unable to distinguish as many colors as you are. Okay, monkeys, about the same as dogs, bulls, mice, horses, pretty much all mammals. But hominids have this extra ability. So how do we explain that extra gene there? Well, we could easily explain a second gene duplication event, right? So maybe there's a gene duplication event here, and we'll make this one green, right? But that explains why hominids have that extra copy of the gene. So we have a gene duplication event here and a gene duplication event there. Now, the last thing, all we have to do is explain why the birds have 4 opsons. And again, we could very easily say, well, maybe there were 2 gene duplication events here. So maybe we've got a gene duplication event here. Right, that leads to that purple one. Again, I'm just going to put these in different order so that they're convenient there. Okay. And then this other one, and we'll make this one yellow just so we can keep up with all of the things. And this is a little bit complex, I know, but it's fairly good example of what's going on. Okay, so we have a gene duplication event there, and then finally, the last one, a gene duplication event here that leads to our red copy, and then another one that leads to our green copy here. Now, hopefully, you're following along. And so this could be the history, right? And that explains it very well. But that's just one of many potential ways. And so it maybe is more complicated than that. And so to do this, what we would need to do is sequence all of these genes, maybe even look for pseudogenes, and then line them up and try and infer the species history, which hopefully we've already got. But if not, we maybe do a whole bunch of genes so it's not biased by only having one gene. And then we do the gene history for this family of genes, which has many duplicates. Now, I'm going to undo all of what I've done, because when researchers have actually done that, when they've gone in and sequenced the genes and analyzed them to figure out the gene history, they get a different, somewhat interesting story. And this is relevant because it says something about the evolution of these organisms. It looks like what happened is actually anciently there were 2 gene duplication events. So we have one there, and then, actually there were 3 gene duplication events. And I don't know that it's really well understood which of these genes. So we could have another gene duplication event right here, or we could put it over here. But 3 ancient gene duplication events. led to 4 ancestral genes. Okay, and so we should probably color code these, right? Just to make it clear like it was before, clear as mud. So here we go. We'll do one of these and make it red. There we go. We'll make this one yellow. And then we'll make this one over here our green. It keeps doing the. There we go. Green, and then finally we've got this kind of blue-purple color. Okay, so 4 ancient genes giving a wide array of visual range where they could see many different wavelengths of opsin. And the birds and other reptiles have maintained that. So we can extend this into the birds, right? Here we go. And do the blue and green also. So what this means is that the common ancestor of mammals and reptiles had 4 opsins, right? Even though we can't go in, they've been dead for tens of millions of years, we can't go in and find that ancestor and look at their opsins. Opsins aren't preserved in fossils, but we can infer that based on the gene history being mapped onto the species history. Now, we're not going to learn the mechanisms of how we actually do that mapping. There's some idea about it in the Svensson paper, but don't worry about the details as long as you understand this level and these what's going on. So let's just extend these out. I don't know why that's wanting to be purple. We maybe just have to change it back to yellow. Yeah, it just seems to want to be purple. Let's make it yellow. Okay. Oh, that's why. There we go. Okay. And then we're going to have our green one here. And then our blue one. Okay. So somewhere along the line, there were 2 of these genes that mutated and no longer became used. And in fact, they've mutated so much, I don't even think they can be identified as pseudogenes. We don't know where they are just because the mutations have made them so different. They've either been lost completely or they've mutated and become so different that they are no longer recognizable as a pseudogene, as what was once an ancient working gene, right? So in this case, the yellow and the green went extinct. Became pseudogenes, no longer were expressed and eventually mutated and were lost from any sort of ability of humans to reconstruct them, even with whole genome sequencing events. Okay, so we then have the continuation of these guys. To account for, I'm going to have to change it just the angle there just a little bit. The red in each of these, yeah, and we have the blue one to account for all of these. Now, we've explained everything. I made this one go a little bit outside. That's going to bother me. Let's fix that. There we go. That's a little bit better. We've explained everything except for why do hominids have 3 opsins. And when we actually do the analysis, we see that this opsin, sequence-wise, is very similar to one of the other opsins in hominids, and it shares a very close history, a close relative with them. And so we actually have another recent gene duplication event in the ancestor of all the hominids, and so they have inherited this midwave that's changed function and gained a new ability and given us a wider visual range. Okay, so this is, as far as our best analyses are able to determine, the true history of the opsins in the amniotes. And so you can see it's somewhat complex, but gives us a really cool insight into recent history. And so now the idea is, and this is, sometimes we don't have good ways to test some of these hypotheses, but we have now ideas about, oh, if this was a recent duplication event in the hominids, why might the hominids need better visual acuity? And some people think that it may have been for being able to look at fruit. Maybe their diet changed and they needed better vision to be able to assess what fruit was good or where they needed to go. Perhaps it was more for visual communication so that they could see facial expressions or flushing of different body parts and that was some sort of a communication between those species. And there are lots of ideas and we don't know for sure, but what has resulted in human and other hominids have better visual acuity than monkeys and canines, but maybe not as good as most birds, because birds have the ability to discern more wavelengths based on their diversity of opsins. So hopefully now you've got a good idea of all of these things. Going back here, make sure you review these ideas: paralogs, orthologs, pseudogene, neo-functionalization, anagenesis, cladogenesis. I want to add one more, just so you know the definition. We're not going to worry about it too much because it's not used a ton. But in the paper, the Svensson and L. Marbrook paper, you will see this term, Isortholog. I think that's how they spell it, or Isorthology. And the idea is that an isortholog is not only an orthologous copy of a gene, but it has maintained the ancestral function. So two genes that are still doing the same thing, the ancestral gene, and they descended from each other at speciation events. And so this is really the power of gene duplication, is that you can have extra copies of a gene, you maintain original function in some of them, those would be referred to as the isorthologs, and then others can diverge and take on new function. And this can be tricky because technically all these opsins have the same general function, right? They're all sensing light. It's just which wavelength they are most sensitive to. And that can change widely, and so there might even be variation. I've just drawn them generally as reds, greens, and blues, but there might be some variation into exact what wavelength the long and short wavelengths see in different species. Now, when we get to near the end of this discussion, we're going to bring opsins back in and look at even more complicated versions. It turns out insects have dozens and dozens of opsins, not just like the handful that amniotes have, and reconstructing their gene history and trying to figure it out in the context of a species history is even more difficult. But for now, just make sure you're familiar with these concepts and able to recognize them on a diagram such as this.